One of the genes that I've worked on, if you alter its activity, plants can get 2 to 4 degrees more freezing tolerance, which is a pretty significant amount of additional freezing tolerance. So our ability to manipulate some of these enzymes involved in lipid metabolism could lead directly to the plants being more tolerant to specific stresses.
Something to Chew On is a podcast devoted to the exploration and discussion of global food systems. It's produced by the Office of Research Development at Kansas State University. I'm Maureen Olevnik, Coordinator of Global Food Systems.
I'd like to welcome back our co-host, Dr. Jim Stack, Professor of Plant Pathology in the College of Agriculture here at Kansas State University. Where does fundamental science and the applied food system intersect? How do students learn to work in a basic research lab or learn that that is really not what they wanted to do? What does technology drive in a lab setting, and where does technology and asking the right questions fit in making important food decisions? This week's podcast helps to answer these questions and to provide a better understanding of food through basic lipid research, interdisciplinary student and faculty interaction, and how this works to improve what we eat. We welcome the team at K-State lipid researchers, Dr. Ruth Welti, Distinguished Professor in the Division of Biology and Director of the Kansas Lipidomics Research Center, Dr. Kathrin Schrick, Associate Professor in the Division of Biology, and Dr. Timothy Durrett, Associate Professor in the Department of Biochemistry and Molecular Biophysics. Ruth, could you give us a little background on yourself and the type of research that you're carrying out here?
Yeah, I'm Ruth Welti. I'm University Distinguished Professor in the Division of Biology. I'm actually a biochemist, and I'm originally from Connecticut, where I went to the University of Connecticut and majored in chemistry. And then I went to graduate school at Washington University in St. Louis, where my training was more on the medical side of biology. And I eventually got to Kansas State, where I initially worked on sort of biomedical problems. But then I was drawn into plant biology about 20 years ago by the opportunity to study the molecules that I've been interested in throughout my career, lipids, in plants. And today my research has kind of two focuses. One is on lipid analysis, and specifically I'm interested in lipid analysis in plants and particularly crop plants. In that work, we use mass spectrometry, which means measuring the molecular mass of the compounds and their pieces to identify the compounds and also quantify the compounds. And then the second component of my current research program is looking at the function of lipid metabolism in plant stress. And by plant stress, we mean we're focusing on mostly what we call the abiotic stresses, which are things like heat, cold, and nutrient deprivation, things like low phosphate, low nitrogen concentrations, and how those are related to lipid metabolism. That's where we are.
Great, thank you. Dr. Durrett, can you give us a little background on yourself?
Yeah, I'd be happy to do that. So I grew up in Zimbabwe in Southern Africa. And so when I grew up there, Zimbabwe, much of our economy relied on agriculture. And when I was growing up, we had a terrible drought, and that really, made me realize the importance of agriculture in our society there. And that was around the same time that genetic engineering started becoming possible in plants. And that was a technology that I became really excited about. So that's why I became interested in plant biology, was to be able to use genetic engineering to improve different aspects of plants. So whether we want to make more drought-resistant plants, that was my original goal. But as I'll describe later, that goal has shifted a little bit. So with that in mind, I came to the US in order to learn about different aspects of plant genetics and biotechnology. So I got 2 degrees, a bachelor's degree in biochemistry at Harvard, and then I also followed that up with a PhD at the University of Missouri. I also spent some time working in a small biotech company doing gene discovery, and that was an amazing time as well. And so throughout that process, my research narrowed in. I worked in a number of different areas, but I did my postdoc at Michigan State plant biology department there. And there I started working on lipid metabolism. I worked in the lab of John Laroghi and Mike Pollard, and they were interested in different aspects of lipid metabolism. And that's where I became interested in the current work that I do now. So what I currently research is manipulating the lipid composition of seeds in order to make oilseed crops produce different kinds of oil that are valuable for different applications. So these could be advanced biofuel applications, or using the seeds as a factory to make different lipid-related products that are useful for various types of industries, as well as always keeping in mind that oil is a valuable food product, ways of manipulating oil composition to make foods healthier. So that forms one aspect. So using genetic engineering strategies to alter lipid composition in seeds. And then part of that, just as naturally involved, as we try to make these changes in seeds, we've realized we don't actually know everything about how plants regulate the kinds of oil they make. And that has impact. So because of that, we've had a number of questions when things didn't work as the way we thought they should work. What are we missing? So that's led to some interesting work where we found some new enzymes involved in the trafficking of lipids and lipid metabolism in seeds. So that's basically where we are today. So some applied work and then some more basic understanding of lipid metabolism.
Great. Thank you so much. Dr. Schrick.
Yes, I think for me, I would describe myself as a geneticist by training. And I think I'm the most fundamental biologist among the 3 of us. A little bit of background on me. I grew up in Ohio, but I grew up in an immigrant family. I'm a first-generation immigrant from Germany. And I think the first time I kind of knew what I was going to be doing the rest of my life, I was about 13, and I read some article about DNA in Time magazine. And I was intrigued, and I thought, this is something that I want to know more about, and I want to do research. And as naive as that sounds, I think I kind of am one of these people that follows my feeling about the future. And I kind of had a plan. I'm going to become a geneticist. I went to Purdue University, and there, that's a land grant university. And the first time I got involved in research, I was a sophomore. And at that time, it was in the early 80s, 1983, Barbara McClintock had just received the Nobel Prize for her work with transposable elements in maize, in corn. And I was intrigued by that story. First of all, a woman receiving the Nobel Prize for her work with plant genetics. And there just happened to be a lab at Purdue that was working with transposable elements, at that time called jumping genes. And I thought, okay, I want to work in that lab and get some experience. And that was my first kind of taste of research and my taste of what it is like to work with plants. Now for my PhD, I decided to work in a simple eukaryotic system. I chose to work with yeast. My idea was I want to be trained in a system where I can learn some genetics and I can work with a fairly simple system, perhaps working with signal transduction. I was intrigued by the idea that there's a signal that's in the membrane. Of course, the membrane is comprised of a lipid bilayer. So there's a signal in the membrane. Proteins are going to transduce that signal down to the nucleus. The nucleus is where the DNA is, and then there's going to be a change in gene expression. And these changes could be occurring in response to environmental conditions or in response to some sort of other change in the life cycle of that organism. So I ended up getting my PhD in genetics from the University of Washington. I worked with Lee Hartwell, who later received the Nobel Prize for his work on the cell cycle. And so I think you might understand, get a feeling for how I value fundamental research. His research in the organism Saccharomyces cerevisiae, which is budding yeast, led to our first understanding that the cell cycle is regulated by genes, and those genes are highly conserved all the way to humans. And those genes are what regulate cell proliferation, the kinds of problems that we have in cancer. But, my love of plants led me to do my postdoctoral work in plant research. And because I always admired the beauty of plants, I wanted to work on development. And again, in development, we have examples of signal transduction pathways that orchestrate the development of a single cell all the way to a beautiful organism. And so I went to Germany for my postdoctoral work, and there I worked in a lab that had done the first saturation mutagenesis screen for mutants that affect development of the embryo. And this was done in the plant model organism Arabidopsis. And so my postdoctoral work, I worked on one of those mutants and cloned the gene and learned a bunch of molecular techniques. And it just so happened that the gene that I cloned that very severely affects development of the plant turned out to be an enzyme involved in a lipid pathway. So that was my entry into lipids. It came completely serendipitously and completely with surprise. And at that moment, that was the moment that I realized I need to collaborate with people that have other expertise. I need to work with someone who does lipid separations and can help me with chromatography separations and mass spectrometry methods to analyze the lipids in my samples. And I need to understand how these lipids affect development. And so since that time, and that was in the early 2000s, it's been more than 20 years, I've kind of chased that question of what are the lipids doing in development. And now I talked about signal transduction. Signal transduction takes us from the membrane to the nucleus. And in my lab, we're working primarily with transcription factors, which are the proteins that bind the DNA to change gene expression patterns to help decide which genes get turned off and which genes get turned on, and at what time in development and in environmental conditions. And so we've been working on some transcription factors that kind of have a direct link to lipids. These are transcription factors that actually bind lipids, that physically interact with lipids. We want to know how that works. Exactly, does the lipid inhibit the transcription factor? Does it activate the transcription factor? At what time? How does the transcription factor even come to see the lipid? Does the transcription factor move to the membrane? Which membrane does it move to? So we have many, many questions that we're attempting to answer in my lab. But I'm so fortunate to be here at Kansas State University with these lipid experts. I've been trying to learn. I always tell my students I am a biochemist in training everywhere that I have been in my career. And I've spent more than 8 years in Germany, I guess, throughout my career trajectory. And before I came to Kansas State, I was at the Claremont colleges, and I had a position at Caltech where I had a bench and access to a microscope. So wherever I've been, I wanted to learn more about biochemistry and lipids. And so I try to interact with Tim and Ruth as much as I can.
Wonderful. That was a good segue into the question that I had coming into my mind. And Ruth or Tim, either one of you can jump in on this, but how do the three of you interact? I mean, we came up with the idea of having this kind of group discussion, and all of you come from slightly different areas, slightly different bends on the way you work with lipids. How does the interaction occur?
So one, I think there's sort of a two-way interaction as I see it. I mean, I'm definitely the person who is the chemistry person in the group. So as I told you, my original degree was in chemistry. Then I went to graduate school in a biochemistry department. My first year in graduate school, I remember just sitting down and thinking like, I better memorize the structures of a lot of lipids because that's going to come in handy someday. And so I did that. And I just sat down and started memorizing chemical structures, which actually is what my expertise is about now. So if people want to analyze things, I think that both Tim and Kathrin come to me to some extent. Tim's had some experience doing it himself before he came here when he was at Michigan State. It's one of the reasons we really wanted to get Tim here is because he also has expertise in analysis. But then the second way is the fact that I am not trained as a plant biologist. And I got interested in plant biology when a former K-State faculty member, Sam Wong, kind of talked me into it, that this would be a great area for me to go into. And it has been a great area for me to go into. But because I wasn't trained in plant biology, I have a million questions all the time. And I basically could not run my lab without Kathrin and Tim to back me up and help me with all the molecular biology and all the biology involved with plants. I love plants, though. I have a huge garden, and I think I should have gone into plants early on, but I didn't. And they're always there, and they've been so great.
Right. I was just going to add to what Ruth said. One of the reasons I was so excited to come to K-State was because we have the Kansas Lipidomics Research Center, which Ruth runs. And because I was using mass spectrometry a lot as a postdoc, I mean, it was critical. This method to like measure the mass of molecules, and when my work involves modifying molecules, we can detect very accurately these changes in them. And so I knew I'd actually met Ruth before I came to K-State. So when I had this opportunity to come here, it was one of the main reasons I came. So that interaction with Ruth is just, we chat a lot about different mass spec methods. And then with Kathrin, our labs have joint lab meeting together because both of us do a lot of, and Kathrin does a whole lot more than my group necessarily, but a lot of molecular biology. So I've always really appreciated coming to Kathrin with questions we have about when we're having troubles with things just not working. She is a great resource to come to and just say, have you tried this? Have you ever encountered this? Or do you have this particular tool? Can we borrow it from you? So to some extent, I feel like I'm in the middle. I use a lot of both methods, and it's great having these experts who can help me out and provide me more information when I feel out of depth with what I'm doing.
Yeah, I would completely agree with that. It's kind of funny because our labs are not next to each other physically. So we're kind of like in three very different locations. Ruth is on the 5th floor of Akron. I'm on the 3rd floor. And then Tim is in another building, but it's a joined building. It's Chalmers on the 1st floor. And so it's a lot of exercise kind of walking between us, but we can contact each other by email and all of that too. But yeah, I just wanted to mention in particular, Tim and I are both working on some genome editing, so CRISPR-Cas9 experiments, and we're kind of sharing some of the reagents, meaning the source of the Cas9, the source of the editing enzyme. And so that's just another example. And I interact quite a bit with students of both Ruth and Tim, and I really enjoy that. I almost feel like it's a family, that whenever there are questions, they will come to me, or I have students coming to Tim or coming to Ruth. There's just a lot of communication between our labs.
Yeah, I was just going to say, there have been times I've met with a student and I said, you should go and ask Ruth this. And they said, oh, I've already met with her. I've already asked her this question. So, and the same with Kathrin as well. They feel really comfortable reaching out. These two great colleagues I have, when they know I don't have the answer, they just go directly to somebody else.
I would say that this is a little unique. I actually visited a colleague at Montana State recently, and he was kind of lamenting how he's there all by himself as the only person who works in the area of lipid biochemistry and modification of crops related to lipids there. And he's pretty jealous of us having the three of us here.
I was just going to follow up on Maureen's question. Clearly, the Lipidomics Center is a unique facility, and I wouldn't imagine there are very many of them across the land-grant system anyway in the U.S. Clearly, it facilitates collaboration here at K-State. Is it also a recruitment of collaborations with other institutions, like you just mentioned, Ruth, with Montana? It would seem like it would bring other people into the larger K-State sphere.
It's a fee-for-service facility, and so people actually mail samples to us, and those samples come from all over the world. I don't really know of any place in the world that does what we do in terms of accepting samples, especially those of plant lipids, from a wide range of labs. Over the years since we started accepting samples, which has now been 18 years, we've had over 700 other labs use our lab. So yeah, wow. So it's a large group of external users. Of course, maybe 40 of those are at Kansas State and the rest are at other places. So we have some labs that, regularly use our lab in their research. So yeah, it is I hesitate to use the word unique, but I think it is fairly unique, if that's a word.
Yeah, that's an impressive number of interested institutions around the world.
Yeah.
When you look at the research scope of that, how broad is that? I would imagine it's an incredibly broad portfolio of what you're supporting.
It's very broad. I would say, so I should be clear, there are other, what we call lipidomics, in other words, lipid analysis facilities using mass spectrometry to analyze animal samples. It's actually something that the Institute of Health supports. They have 4 large facilities that support metabolite analysis, which includes lipids, and they fund them very nicely. And so for animal samples, there are a lot, but I would say still about 2/3 of the samples that we receive are from plants, and about 1/3 are from other organisms. And those other organisms include everything from animal samples to protozoan parasites, yeast, bacterial samples. Yeah, I'm not sure about archaebacteria, but a wide range of organisms. And I think that's kind of where my previous experience working with other organisms and other systems has been helpful, that I'm pretty familiar with bits of quite a wide range of organisms. And I've become over the years familiar with plants ranging from simple plants like moss all the way up through dicots, basically.
Yeah. So maybe all 3 of you, but certainly 2 out of 3 of you in your introductions, you were mentioning the importance of lipids and lipid biochemistry to stress in plants, how plants manage stress. If you just look at the weather profiles, the climate weather profiles of the last, say, 10, 15 years, and the climate projections for the future, and our increasing dependence on plant systems for, not the least of which is food security, but other aspects of human health and well-being. What do we need to know? What are the big questions that we need to address so that we can actually breed perhaps plants to more effectively handle this stress that they're facing now, but it looks like will increase significantly over the next decade or 2? What are those big questions?
I think one big question, I'm sure Kathrin and Tim can add to this, is that we need to know what genes are involved in the responses to stress in the area of lipid metabolism. And there are over 1,000 genes involved in lipid metabolism. And one of the big challenges in the plant system is that there are some groups of genes involved in lipid metabolism, particularly those are involved in lipid hydrolysis, that have a very large number of genes. For example, there's over 300 genes involved in lipid hydrolysis, which means like removing a fatty acid from the compound it was originally in. And so those genes are expressed in different locations within a plant, and that's different tissues, different cellular locations. That they're also expressed at different times in development, and they're also expressed in response to different stresses. So you can imagine with 300 genes that could theoretically carry out the same reaction biochemically in a test tube, identifying like which ones are important in a specific stress catalyzed process. So something that you have a stress, and then which genes are acting, and where are they acting, and what are they doing, and what are they acting on? All those kind of questions are really, there's a whole lot to still do in that area in order for us to be able to understand how those relate to the physiology of the plants and the way they respond to the stress.
Yeah, and I would add to that, we know very little about where the lipids actually are in terms of subcellular, because many of these analyses just kind of take a leaf, grind up the leaf, and then what's the lipid composition or another part of the plant, but we don't know, like, are these lipids in the epidermis? I'm particularly in our research, I'm interested in the epidermis. That's the L1 layer. It's like the skin of the plant. It's the first line of defense. The epidermis has hair cells that have, some of these hair cells may act like trees in a forest, so that you don't have, so that you can kind of collect the dew at night. So that offers a cooling to the plant leaves or other structures. And the epidermis also contains the guard cells, which are the cells that, either open or close for so that plants can breathe, basically. So understanding the lipids that are present in the epidermis and the genes or proteins that act on those are going to be really important. But that's true for other tissues inside the plant as well. And so we're really kind of at the beginning of being able to analyze plants at that level to know in which cell type exactly the lipids are and how that changes over development and in response to different stresses. So I think that's one area that it's a tough area to look at right now because we need to have some sort of imaging analysis that is kind of an in situ. There are these in situ experiments, where you can look at RNA. Where is RNA inside of a leaf? You can now do these single cell omics experiments, but single cell omics for lipids is not really quite there yet. We've got single cell technologies for RNA, for other omics, but in terms of looking single cell level, that's something that is going to be a technology that is needing to be developed in the future.
I was going to add one thing. It's like the one reason that it is particularly challenging is that unlike a lot of other processes where genes are only expressed, in other words, where genes only make proteins when needed, a lot of these lipolytic enzymes are present at a pretty constant level. And their expression isn't necessarily regulated in response to the stress. The enzymes are activated on a protein level in response to the stress. So that there may be translocating from the cytosol to a membrane upon the stress, or there may be interacting with some other factor that activates them. So they're there all the time. So just looking at gene expression isn't really enough to help us understand what enzymes are doing what. So for that, we focus on looking at mutants where we've knocked out that enzyme or knocked out the function of that enzyme and related enzymes to try to understand what's happening and how that affects the plant's physiology and its ability to handle stress.
We've got some very fundamental science discussion going on here, which is awesome. Where does this fit in the food system? What is the impact of what you're doing in the overall food system?
So I think one of the genes that I've worked on, if you alter its activity, plants can get 2 to 4 degrees more freezing tolerance, which is a pretty significant amount of additional freezing tolerance. So our ability to manipulate some of these enzymes involved in lipid metabolism could lead directly to the plants being more tolerant to specific stresses.
Yeah, I was going to suggest an idea like that as well, that by understanding genes involved with abiotic stress, we can breed for those. So those can be used as markers. So we can breed varieties, or we can use biotechnology approaches to make plants more tolerant. Another potential application through understanding lipid metabolism has come from the development of high oleic soybeans, for example. So by understanding pathways that make these different fatty acids, we've been able to now specifically target genes that regulate the fatty acid composition, and thus the health profile of lipids in seeds. So there are a number of products on the market now that should be impacting consumers that have been developed through an understanding of lipid biochemistry.
Yeah, another example would be aging of seeds, that manipulation of reducing lipid hydrolysis in seeds can make them age better. And so they can be more stable, like the oils in the seeds can be more stable.
Yeah, and the transcription factors I had mentioned in the beginning, they are involved in biomass. So there have been certain of the members of the family, if you express them at higher levels than the plants, and this has been worked out in a number of crops as well, including rice, that if you express those proteins, you'll get a plant that is of increased biomass and also more drought tolerant.
Are the industries that support plant-based agriculture engaged in these types of research projects? It would seem like ultimately that they're going to benefit from this research. Are they contributing to this effort yet, or is this still too fundamental?
With regard to, for example, the high oleic oil, that has had a lot of heavy industry involvement just because that is a large commodity crop. So it's been done in soybean. I know they're trying to make even higher oleic canola and sunflower as well. So that has had a large industry involvement, and they're using different technologies as well. So initially they used gene engineering to do that. Now they've actually found additional ways, non-transgenic ways of manipulating oil composition as well to make high oleic varieties as well. That all has involved heavy industry involvement there just because it was such, in some ways, an obvious target to go after.
Also, when we first started doing lipid analysis, we at that time had quite a few samples from industrial, like plant biotechnology labs. We've had a lot fewer in recent years, which my take is that they basically started their own facilities that can handle the analysis using the same kind of technology that we're using.
Opportunities for students, certainly in terms of graduate research programs, but what about internships for undergraduate students? Are they part of the program at the Lipid Center?
Well, in my own group, I've had as many as 8 undergraduates at one time. Right now, we only have 3, but yes, they're definitely a part of our program. And maybe Tim and Kathrin can speak to that also.
We all have undergraduates in our labs, and they do a lot of great work. I think they all come out, at least in my lab, coming out of knowing how to do some kind of lipid analysis. Sometimes that is with the Lipidomics Center. Sometimes it's with instrumentation we have in our own lab. So, and hopefully that gives them a sense of they can both do molecular biology as well as biochemistry as well. That helps them figure out what they'd like to do, and then hopefully guides them with where they want to go in the future.
I should mention that I also teach a class in the fall. It's a CAT community, and it's called Exploring Scientific Research. And in this class, I have 21 students, and I identify labs for them to join. They're first semester freshmen. They have no idea what they signed up for at the beginning. That's what I found. Like last semester, one of the students joined Tim's lab, and she, I believe, is still working in Tim's lab. Yeah, so in some cases, these students it may be a 1 semester event for them, but certainly a number of the students continue to pursue research in the labs that they got into through this class. But I do think this is a great opportunity for new K-Staters to get involved in research.
Yeah, we certainly all have undergraduates. I'm going to be having 4 undergraduates working almost full-time. 3 out of 4 will be working pretty much full-time over the summer. I have seen cases where there are students that come to the lab as undergraduates wanting to go to medical school. And in one case, this person was highly accomplished, got a Goldwater scholarship, and he ended up going into research over choosing medical school. He just kind of caught the bug. So I think the opportunity to work in a research lab is something that can be really life-changing.
I think those undergraduate opportunities are really important. I think it helps a lot of students decide where they're going next after they complete their degree, and to get a taste of research and find out, wow, I don't want to do this. It's better to find it out there than once they've enrolled in a graduate program. So I think those opportunities you're providing are outstanding.
Yeah, I would totally agree that it's equally valuable to find out this isn't for you.
Yeah, I do some opportunities for undergraduates up at the Biosecurity Research Institute to teach them about working in containment. And the overwhelming majority conclude, I don't want to do this for a living. So yeah, that's an important opportunity for them so they don't kind of get down a track that they really aren't suited or pleased by. I've got a really philosophical question. So you're all really into technology, and technology has had a very interesting relationship with science for the last, say, 200 years plus. And science has a question, and they need technology to answer the question. So the technology gets developed or refined, which allows the science to ask either more questions or questions in new ways. And then kind of this feedback loop where one drives the other. But it seems in the last few decades, technology has played a greater role in deciding where we go in science and what questions we ask because of the available technology. Is that just my imagination, or what's your experience with that push and pull from technology and science?
I think that's a great question. I think as somebody who went to college and calculated their chemistry exam with a slide rule, and basically even in graduate school, I was like pushing the buttons on a calculator to determine the quantities of the fatty acids in my sample. It's really, to now I'm measuring 400 things simultaneously on 5,000 samples. It really is a case of where things are possible that we couldn't even have imagined. But I can also say that in the case of the actual impetus for lipid analysis, using technology like mass spectrometry, obviously that was driven by the need for the analysis in science. But then again, it does advance the science in ways that you couldn't have even imagined. I think it definitely goes both ways.
Yeah, I mean, we are limited by what we see, most definitely, but I find it challenging to someone ask me, we've got this new technology, what can you do with it? I mean, I'd rather have the questions and then look to see which technology I can use to answer that question. I mean, there are certain technologies like the genome editing, which is going to be beneficial to the field of lipids as well, because we have many genes that may be small genes. And if we can make mutations and they may not have already available mutants that we can analyze. So if we can make those mutations in those small genes and see what the consequence is on the organism, that's very valuable in terms of that technology. That's why I feel like I'm a molecular biologist, biochemist in training all the time, because there are all of these new technologies that come up as we progress over the years. And that's what I love about it too, of course. So we follow the stream and then we continue to ask our important questions.
But I think that for all of us, our affinity for working in a model system like Arabidopsis thaliana, which is the model system for plants in general, is due to the fact that there is so much technology associated with that particular system, because there are big libraries of mutant available, right? That we can so we are letting the technology drive us to some extent. That's why we're not working in some really exciting rare plant.
I might push back a little there, Ruth. I mean, I agree with you. I think there's a reason why so many people use Arabidopsis. And I remember when I started my postdoc, that was the first time I worked in a lab that didn't use Arabidopsis. I was kind of lost. But then technology, like there was one, I mean, this was when the development of next generation sequencing came along. And so for the first time, you could obtain like gene sequences of non-model organisms. And that completely revolutionized the project that I was working on and really like launched my career. And since then, we've continued to utilize that tool. And it's been amazing how next generation sequencing has advanced so much since 20 years ago now. It's just amazing. Kind of what Kathrin said, it keeps constantly need to keep learning these tools and new methods. But I would also say, I still think as much as technology drives what we can do, it also comes down to the questions that we're asking. So, I mean, I think there's always a temptation, I'm just, I can use this new technology and I'm going to get all this information. But I think if you're not asking the right questions, you can do a whole lot of work and not get a whole lot out of it necessarily. That's something I always challenge my students with. They learn about a new method and like, we should do this. And I'm always like, that's great. But what are we trying to answer with by doing that?
I mean, I think in one sense, you're arguing the same thing. I argued that the technology presented new opportunities. So we went for that, but it still was the technology that drove us to some extent. I do think that one thing that's happened is that the technology has allowed us to do discovery projects where we didn't have to have a question in place at the beginning. I mean, or our question could be very broad and not a very targeted question. Like we can use things like genome-wide association studies to get a list of candidate genes that are involved in lipid metabolism by doing the chemical analysis across a wide range of plants that are related and that we have sequenced genomes for. So I think it's opened up opportunities for us to focus our questions, maybe to get more questions that we couldn't have gotten without the technology.
Yeah, that's good. Thanks. Yeah.
Jim had asked a question about industry funding or industry support of the work that you're doing. Just looking at it maybe from a little bit different angle, how is this information transcribed or moved out into the useful community? Those people that are going to take all of that basic research that you've done and use it to do something more with it. You know, the industry supported stuff that you described, Tim, I get that. And they've identified an issue and are taking it out. But there are a lot of things that probably are a little fuzzier than that. How do you get that information out to the people that will do something with it? How is that bridge crossed?
So I mean, part of it is when you make a discovery, you publish your results, and that's then available in the scientific literature. And I know because I work primarily with the model plant, Arabidopsis, some of the discoveries that we've made in that organism were later confirmed or found to be similar kind of stories or similar proteins, similar lipids involved in crops like tomato, like cotton, like rice, and others. And so I feel that just doing curiosity-based research, basic research, will put information out there so that when someone goes into their wheat genome and they find a gene, they don't have to guess what it's doing. They can do, based on sequence similarity, they can say, oh, in Arabidopsis, this gene is involved in hydrolysis of phospholipids. And so we've embarked upon a lipid-related phenomenon. And, without that basic research having been done in a relatively simple model organism, Arabidopsis, there would have been no way to make that conclusion or to come to that kind of understanding of what's going on in the crop. I guess it's pretty much in terms of trying to contribute to the general knowledge in the scientific community.
I just know that, so I'm with Ruth. Here's my circular slide rule. Back in the day, I keep it here as a reminder of how fortunate I am now. And if somebody told me when I was in grad school that I'd be sequencing the genome of a bacterium for about $200, you'd laugh out loud. But here we are. I think it's great that you have your lab groups meeting together that are perhaps bringing a different perspective. I wish we did that more often. I think to the benefit of the student, just to hear different perspectives. I mean, you can look at an organism and really see it through different lenses. So I commend you on that.
I think that we've collaborated directly with people in some of the more applied plant sciences, such as agronomy. For example, I've worked with Vera Prasad quite a bit, and to try to better understand the role of lipids in heat tolerance in crop plants. I know Tim has some collaborations in agronomy also, I think. So a lot of it is direct interaction between the labs.
Yeah, just again, to echo what Ruth was saying, I have some great collaborators in agronomy. Ignacio Ciampiti is a great example, and I've appreciated working him and others just for their very different perspective of how to manipulate yield and plant production, right? So they have their agronomic focus. What unifies us, though, is understanding like how is the plant responding at a molecular level. So they see these big effects on yield, but they're at the bottom of it, they're asking questions about how is that being caused at a molecular level. And by understanding that with the help of a biochemist such as myself, maybe we can better identify plants that would be able to increase yield or drought resistance or abiotic stress resistance.
Yeah, I'll add also that all three of us belong to a group called Lipids of Crops, which is USDA. It's related to our agricultural experiment station appointments. It's the USDA multi-state group that started with the states in the Midwest, but it's basically all scientists who are interested in lipid research, and it's a collaborative group. And some of the people in that group are quite a bit more applied than we are, but we have collaborations with them. Like I have a collaboration with Shruti Narayan at Clemson, who's an agronomist and works on heat tolerance. They're right now planning a big genetic analysis to be able to understand the important genes that are involved in heat tolerance related to lipids in soybean.
So most of our conversation today has revolved around the abiotic But I think I noticed in the NSF funded project that Pseudomonas syringae was also listed as a stressor, and someone who has a personal interest in P. syringae. I'd love to hear more about.
I've worked on a collaborative project with Jyoti Shah, who's at University of North Texas, looking at lipid changes in bacterial infections. There's actually a group of lipids that I'm particularly interested in that are super induced by bacterial infections, and particularly by avirulent interactions. And they're much higher when there's an avirulent interaction between the bacteria and the plants. In other words, an interaction where the plant's fighting back and it's not actually killing the plants, than in a virulent interaction when the plant is actually dying from the infection. So we're kind of interested in these lipids and what their actual role is in those biotic interactions.
I'll just mention, so my first research experience was in a plant pathology lab where we were looking at interactions between different pathogens, so molecular interactions between different pathogens and the model plant Arabidopsis. And one of those pathogens was Pseudomonas syringae. I remember spending a lot of time infiltrating leaves with different concentrations of Pseudomonas. And so it was primarily a genetics-focused lab. So we were interested in identifying genes through mutagenesis that affected interactions with different bacteria, as well as some fungi. And what's been remarkable, so this was all pre-genome days, right? So we didn't have the genome of Arabidopsis. What's been remarkable over the last 20, 30 years now with this technology we've talked about is how many of those genes I was working on as an undergrad are lipid related. So it's been this really nice full circle where sometimes I run into people at conferences who I remember as undergrads and they're working on lipid related genes. So that's been kind of interesting to see.
I think it's also interesting to see that a lot of genes that we don't call lipid related are actually lipid related. I think it's been one of the big things that's going to come out of these genome-wide association studies is that here we're looking at genes that are associated with changes in specific lipids. And less than half of the genes that we found when we've done those kind of studies are genes that we would have originally thought of as being lipid related. So some of that could be noise, but I think a lot of it is actually pointing us to discover things about those genes' function that we don't know yet. There's a lot of follow-up that we need to do.
I think that's the name of the game with research, isn't it? There's always a lot of follow-up. I would like to open it up, see if there are any finishing comments that any of you would like to provide, if you've got questions of us at all.
It's been fun.
Great. The center that you are operating, I think the word unique was used a couple of times. I think it's just phenomenal. It'll be great to get the word out, let people around the world know what's happening here, and some very exciting basic research that's going on. I really appreciate it.
Yeah, I want to say thank you. I learned a bunch and enjoyed the discussion a lot. Thank you.
If you have any questions or comments you would like to share, check out our website at ksu.edu/research/global food and drop us an email. Our music was adapted from Wayne Gowen's album, Chronicles of Carmela. Special thanks to him for providing that to us. Something to Chew On is produced by the Office of Research Development at Kansas State University.